Okay, we are just about at 9:25 Pacific time. I'll just give everyone just a couple more minutes to rejoin us for those that have stepped away from their computers. And while we wait just a couple more minutes for everyone else to rejoin back, next we'll be having Randi rejoin us, and she will present accessing the data, query reporting and examples. So as I mentioned, Randi has a wealth of experience with the database, one of our core team members in the IEDB team that helps facilitate the interactions from the curation piece to actually you, the users, where you're querying and getting the results from the database. So Randi has spent a lot of time reviewing your questions that you submitted. So we had a pre-event survey that was sent out to all of the attendees, and we had over 100 questions submitted. So Randi did a wonderful job in reviewing all of those, categorizing them, and then responding to them within this next hour that she will be presenting. So as I mentioned at the start, you might see a couple of questions that are familiar to you. Some questions will answer multiple aspects of the database and all of its key features, and some will form the key example that she'll be discussing as well. So I think we will just wait one more minute. I can see a couple more people are rejoining. Okay. And I see we've got one more question that was submitted a little bit after we wrapped up the Q&A with Alex and Randi before. So we'll save that question for our next Q&A, definitely. Okay. So I think I will slowly wrap up. That's everyone rejoining. Thank you all very much once again. That concludes our short break. And now I would like to formally introduce Randi back to the screen where she will kick off accessing the data, query reporting and examples. Thanks very much.
So you ready for me?
Absolutely. Thanks, Randi.
Okay. So as Nina mentioned, I'm going to use the examples you gave us to run through how you query the database. So this is the homepage, and as mentioned, this is where you can do to 99% of all the queries that people do in the IDB. These 6 search panes were because this is how people want to interact with the data. Some people are very interested in just specific peptide, and some care more about a disease. But these 6 panes capture what most are interested in. So I'm going to walk through each of 6 panes and show you some features for these type searches. So the first one is the epitope search pane, and in it you can use the radio buttons if you want all linear, all discontinuous, or all non-peptidic epitopes. But one of the common ways people search is they have a particular peptide. They want to see what data we have for that peptide. And you can add to the box, the text box, and whatever peptide sequence you have. I'll point out that you can search match, which will find data for exactly the same epitope that you have. But it is very common for other authors to research a slightly different peptide from it may be 1 or 2 amino acids longer on one end or the other or both ends. It may have a few residues slightly. Sometimes that's meaningful because they're different isolates of the same protein, or it could be that they just already had that on hand and it was so there's not always a serious meaning to why it might be slightly different, but if you don't get a lot of data, you think there should be the searches to search for a broader range of epitopes. So if you put in your peptide, search for 90, 80, and 70% identity blast matches. Or for substring, you can put in a longer sequence, and we will tell you epitopes within it that we have data. And then the next search on the homepage is the antigen. So this is if the epitope is natural, what organism did it come from? What antigen did it come from? It's a bigger thing. So for the organism search, you can just type into that search box and you will get, using those NCBI taxonomy synonyms and term names, the best hits. And you can scroll through these hits if there's a lot of them. If you've typed something that's really common, like the word virus, you're going to hit Here I showed that by typing ORO, I get coronavirus as the first hit. And then the second search box in the antigen is the name. So this would be like the protein that the epitope is from. And so if you have a protein name, you can type it here and you will see all the hits. But I want to warn different species have proteins with the same name. So say many mammals will have an insulin protein. This will have a capsid protein. This isn't the most efficient way to search because those names are that unique. And if you have a truly unique name, you can enter it, of course, but an easier thing to do is to enter the organism of interest and then run your search, and we'll present to you that reference proteome for that species and show you which proteins we have data for from that species. And then the next search box is the host. This is the source of the effector cells or T cells, the antibody or the MHC molecule, or the antigen presenting cells that processed an epitope for ligand dilution assays. So you can use your buttons for the most commonly searched upon hosts, being mice and non-human primates. But if you're interested in any other host, you can use this free text box and just type in it, or you can click the find button, and that find button opens the finder that presents to you that NCBI taxonomy tree I was talking about. So these finders show you those ontology hierarchies that you can click through. You can search it by typing, or you can click open the boxes and navigate to see what we have data for. And so we have all hosts that there's published epitope data for. So a cat, it could be a camel, shark, any species that there's data published for, they will show up here. And if a new species is in next week's PubMed query, it will then be added to this tree. And I did a little search to show you. I typed the word macaque and hit search, and it gave me 7 records. And I want to point out that the green boxes with the plus sign add that value to your search. Once you've added it, it shows up at the top. And these are multi-search boxes, so you can add more than one. But I clicked on the yellow highlight where it says T and there's a yellow highlighter there. You click on that, it opens up the tree and shows you within that NCBI hierarchy, where does the organism I clicked on show up. And this is useful if you want to see what siblings we also have data for. So here you can see all of the macaques that we have data for. And you may decide you want to look at 2 of them. You could click both the crab eating and the rhesus. Or you may decide I want to see all of them and I'm going to go for the higher node. By presenting the data this way, it gives you an idea of all the species we have data for. And then you can also see the synonyms below and link outs to those ontology identifiers. Then now on the right side of the homepage, the first search box is for the assay. And in the assay search pane, our default value is all positive assays. And this is because that's what most users have tended to want. But if you want positive and negative, you unclick that, and now your results will include both positive and negative data. If you're interested in all assay types, you can leave it as the default and just hit search. But if you want to narrow your results to just a T cell, or just a B cell, or just MHC ligand assays, you could unclick the ones you don't want. Or if you know you're only interested in a very precise assay type, you can enter that here now. You can type IL-2, you can type neutralization, and it will auto-populate based on the assays we have that meet that criteria. Or like the others, you can click on the find button. And that opens the assay finder. So this presents to you that OB ontology, the ontology for assay types, and lets you navigate the tree to see all the different assays for which we have data. And here I've opened a B cell assay a little bit, so you can see some of this. And here I've opened T cells, so you can see if you're interested just in cytotoxicity, you can check that. Or if you're interested in 3D structures, you can check that. So you can navigate through, and this will give you a really good idea all the types of assays we have data for in the IDB so far, and this may change how you decide to perform your query. And then the next search pane on the homepage is the MHC restriction. So for T cell assays, if the authors provide MHC restriction, and we capture it, and for MHC binding assays and ligand dilution assays, there will always be a MHC molecule. But the level to which that molecule is described is entirely up to the authors, but they're all grouped together using that MHC restriction ontology. So all at class 1, class 2, or non-classical MHC molecules may be searched using these radio button. But if you have an exact MHC that you're interested in, you can type it here. And if you just start typing a little bit, it will autocomplete with all of the best hits. But if you click the find, it opens up that ontology view showing you the finder and application, which you can navigate through to see which MHC we have data for. So this gives you an example of many of the species that we have: horse, duck, dog, clawed frog. And this totally reflects the literature to date that we've encountered. So these are all of the species we've encountered some MHC information in the literature for. And I opened cat just because it was a smaller branch. So cats will have more than one class 1 allele, but this is the only one for which we have data so far. And if you ever think that there should be more information and we've missed it, please let us know. We always want to hear if there's something we need to curate and we haven't. And this tree lets you search from the levels of class 1 as you could with this radio button, but you can now here narrow it certain species, or cattle class 1, for example, or I want all HLA-A or FLAE, for example, or you can pick the precise MHC molecules that are important to you. I'm using the tree or by typing. This is the last search pane on the homepage. It's the disease. So this is the disease in the host at the time of the experiment. So did they have a relevant disease that led to their immune response to the epitope? So did they have an infectious, allergic, or autoimmune disease? We have a radio box because these are the most common search parameters people care about. But if you have a specific disease, you can type it in this box. And again, using the disease ontology, we'll use all of the synonyms to auto-populate based on what you type. Or if you want to just browse and see what sort of data we have, you can click the finder and it'll show you the disease ontology tree view in the finder. And here the diseases are organized by the higher levels of allergic, autoimmune, etc. But if you click them open, you'll see how they're further grouped. So all metal allergies are together, all respiratory allergies are together, drug allergies. Or you can pick the higher node once you see what everything is in there, or you can click down to exactly what you're interested in. And the user submitted query that I used for my first example was, how can we use IDB to do research on COVID-19 immune responses? So in order to search for this query, all I did was on the homepage, I went to that organ box because these are epitopes from COVID-19. And I started first with a vaguer query than what asked, and I in general advise people to do that to see the breadth of information we have, and then you can further narrow once you see what we have data for. So I CRRO, diploid coronavirus, and I selected that. And that was the only thing I changed from the default search. So then I hit the green search. Just to let you know, because you can build your query, you can pick as many features from each of these 6 panes. I may have wanted to pick a specific host, or I may have wanted a specific disease or MHC molecule. So the search doesn't know when you're finished building it. So once you've made all your selections, you then hit search. And then I got this result page showing 3,700 epitopes from coronavirus, and this is all positive assays only. And then I'm going to walk you through what the results look like, how we present them to you. So over on the far left, these are additional filters, which we will get to how you can further narrow your results. At the top are the you've applied so far. So these are the only 2 search criteria I added from the homepage, positive and coronavirus. If I wanted to change my mind now, I can X them off and get rid of them. And then the next part of each tab, we have epitope tab, antigen tab, assay tab, receptors tab, and reference tab. And this is because each of these types of data have slightly different column headers, so they're presented on different tabs. They each have a separate export also. So each of these tabs has a count of how many rows are on that tab. And so for the epitope tab, we have 3,700 epitopes. And then each of these results tables has a table header. And the table header has these little down arrows next to them because each column is sortable. If you click on epitope, it will sort alphabetically. If you click on number of references, it will sort numerically. And each of these tabs has that same feature. And then on all of the tabs we have on the far left, a details or an ID column. And that gives you the stable ID for that row. So this is the ID for this epitope. And then each of those is clickable, and it takes you to details about that epitope. So we, again, store a great deal of information about all of our epitopes and all our assays, but make these table views easy to understand and not overwhelm you. So we hide a lot of the information, and this is just a summary that I presented to you. But when you click on the details, you see everything. And when you click on the export, which I'll show you, you get everything. So we allow you to export and see everything, but we consolidate it into a simpler view for the result page. So here I clicked on one of these details, and this is what an epitope details page looks like. And this is that same ID that I clicked on. And we give you a textual summary that sums up all the information we have for the epitope at that time. So we add new data every week. So we may add additional publications. This epitope was tested in 11 publications. So all the data from those 11 publications is summed here all together. If there were B cell, T cell, MHC assays, we list assay types, and then we let you know how often they were tested and how often they were positive. So this is something that comes up as a common question of what's the restriction for an MHC molecule, for an epitope. So an epitope may be tested many different MHC binding assays, T cell assays, ligand dilution assays, and the conclusion of what its restriction is isn't always 100% clear. It may have been tested in multiple species. It may have been positive for more than one MHC molecule, or it may have been highly positive for one in particular, and then sort of weakly positive to some others. So we compile all the data together and as the authors presented it. So as many MHC molecules as for which an epitope was tested, we present to you whether they were positive or negative. And the same is true for all the T cell assays, all the antibody assays. So if every paper tested an interferon gamma assay, we would add them all to the database. Whether they were positive or negative would always be included. And then back to the results page. So these were the epitope details I had just clicked on. I also want to point out the export, that you can export each of these tabs, and they each have a separate export that gives you all those same details that were in that detailed view. And the formats is with and without IRIs. And the IRIs are those ontology links, those identifiers to those external resources that I've been showing you. And when I'll go through it later on, and I'll point out what they look like. And then on each of these tabs on the results page, we also have these little funnels with the plus sign. And this lets you filter your data further right there on the fly. So if you want, you see all the epitopes we have data for, but this one's studied, so you want to just focus on it for now. You can click this little funnel and it will filter to just that epitope, and it will appear up in your current filters. And if you change your mind and you want to go back to where you were, you can exit back off and research, and you'll get back to where you were before you added that additional filter. And this is true for the antigen names, the organism names. Anytime you see the little funnel, you can then filter to specifically that concept. And then the next tab over from epitopes is antigens. And when you first go to the antigens page, there's this pop-up telling you about the immunome browser. And this is because this is a really important and prominent feature that people like to use and are often looking for when they go to the IDB. But when you exit out, it won't come back. So I exit out so I could show you what the antigens page looks like. And again, it has the blue header that's sortable, and you can click on each of these little arrows. And then there's the icon for the immunome browser. So if you've closed it out and you want to get it back, you click on that, and it will show you what your query was. So in this case, my query was just positive assays from coronavirus. So this is that's data we were just looking at, but for this specific spike glycoprotein, because that's the one whose little graph icon I clicked on. And I'm going to walk you through the entirety of what's on the immunome browser when you go to that page. So when you open it, the first part shows you the response frequency. So this, out of the number of subjects tested, how many were positive. So if an author gives us that information, we always capture it. That's in the details. But if they do not provide it, we count positives as 1 out of 1, and we count negatives as 0 out of 1. And this information is all available to you as a table that I'll show you. So the query you provided is here. If you want to change how the graph looks, change your query. So this is, to date, all the information that we have for all coronaviruses plotted on the spike glycoprotein. And then the next part of the immunome browser, when you scroll to the page, shows you the epitope assay count. As I showed before, it'll have red, the negative assays at that region. So amino acid 1 through 67 has both red and blue, blue being positive, red being negative assays. And assays performed. And Alex mentioned the negative data. This is why we want to put that negative data in because we want to be able to show you that it was actually tested versus nothing, the proteins where there's no information for certain regions. And here I zoomed in a little bit so you can see how the data overlaps. And that's because this is all data. So if you selectively looked at antibody or B cell only or T cell only, you're going to get a different graph or a different species, or just in disease cases it changes. And one of our user queries was about, do epitopes go away, and how do we clear out epitopes that aren't true epitopes anymore and just repopulate the database. And we don't do that. We accumulate all the data from all the papers that we ever have curated or encountered in literature. And it's up to your query to determine what are the epitopes for you. So both red and blue areas. And that doesn't mean it is an epitope or isn't an epitope. It means that the epitopes tested in this region meet the criteria of my query. So if I wanted to only look at antibody, I may get a very clear spike that this is a very strong antibody epitope, but it's not a T cell epitope. And both of those data points need to live in the so that you can draw those conclusions. And then the last part of the Immunome browser, when you open it up, is the data table. So these are all the epitopes that were put onto the graphs we were just looking at. And I want to point out we do have discontinuous epitopes also. And we show you the positions epitopes map to, how identical they are to the reference proteome protein. Because sometimes the reference proteome protein differs slightly from the epitope, the protein these were originally just to let you know. And then if the authors gave us number of subjects tested, you can tell, like this was 17 out of 42, where 1 out of 0 was probably the authors didn't provide it. It was just a negative assay. And then all of this information is exportable, and you can use it in your research, and you can use it in figures and papers. So then the next tab in the results page, we looked at the epitopes, the assays, and now the antigen assays. So the assays are usually what most people want to look at. So in the assays tab, it's further divided into T cell, B cell, and MHC ligand assays. And that's because these have different column needs, different types of information. MHC restriction column is both in the T cell and the MHC tabs, but it's not cell tab, for example. And we capture a summary of information about each assay here that's presented very quickly in this small table. But again, you get all the details when you click on the details link and when you do the export. And so we repeat information about the epitope, the host, the immunization situation, what was the assay antigen for this row for that particular assay. If there was restriction information, it would be here, and a small description of what the assay was. So each of the tabs have that same lineup, and they all have the same header with the sortable columns. And then we still have those little funnels with the plus sign. So if you want to filter, you see a paper that's really interesting to you, you can filter for that reference here. You can filter for the epitope. You can narrow your results to a particular host. That's the host you're going to be studying, and you want to see what data we already have for that same host. You can filter onto the MHC restriction. And once you click any of these, they'll show up on the header row as one of your filters. And then now I want to show you what the details look like for any particular assay. So you saw a little bit of that in the video about the curation. From the curation standpoint, how we were adding those details into the curation. Now this is how they look when you see them on the external site. The details, a link is again on the left side, and you click on the ID. And that's the stable ID for that row. And the assay details page, regardless of the assay type, follows the same format. The first part of that page will always give you information about the reference, where we got that experiment from. So the data comes from the literature. This was the journal. These were the authors. This is the title of the paper. These are the link outs. These are the IRIs. This is a link out to that paper. And then this is the date that that paper was last changed in the IMDb. And then each of those assay details had the next section for all of them will always be about the epitope. Which epitope is that assay specific to? So it's about either this epitope being tested or a receptor that's specific to this epitope. So this epitope in the particular paper we're talking about for that row was described as being from this specific strain of beta coronavirus 2 by those authors. So we're true to the paper. We're curating each individual paper as its own entity. And in that paper, this peptide was stated to be from this strain. And then here, these are the link outs for that strain. And for each one, we tell you where we got it from the paper. So this epitope came from figure 6. And then in the results page, on the assay tab, the details next section after the epitope for antibody and for T-cell assays is about the immunization. There's a section on the immunization. For naturally processed or MHC ligand elution assays, the next section will be about the animal, if there was an in vivo processing step. So with the APCs that were processing the epitope, was there immunization going on in that individual? For T-cell and antibody, was the host immunized? So in this case, the TCR transgenic mouse was injected with the epitope itself. It may have been a human who had the disease, and that would also be described here. So you can see exactly the details of how that immunization took place. And if there's additional information, the curator would add comments. And then for antibody and T-cell assays, the next section will be about the effector cells or the antibody itself. So in this case, this experiment used lymph node cells taking direct ex vivo. If there was an in vitro restimulation step, that would also be described. This assay was a proliferation assay. So that is described here in the OB ontology identifier, that's the IRI, that you can link out to read more about the assay type if you're interested in that sort of thing. And in this case, this epitope had a restriction as stated by these authors for this paper. Another paper may decide a different restriction for the same epitope. But we're curating here individual paper. And as you can see in the antigen, so this is the last section for any assay details, regardless of the assay type. It's about what was the tested antigen in that assay. And in this case, it was a proliferation assay in response to the same peptide that is the epitope. But this data was taken from figure 6 of that paper. And now back out to the results page. So we went through epitopes, antigens, and assays, and the next tab over is the receptors. So these are antibody and TCR sequences. And for coronavirus, as you can see, we have a large number of TCR sequences. At the time that I made the screenshot, 15 antibody sequences. And just like all of the different tabs, we have the blue header bar that is sortable. So you can click on the arrows to sort. And then we have on the left the identifiers that you can click on to get more details. So you can see that these IDs and the funnel also is the filter. So I'm going to show you what the details look like for a receptor. Receptors are grouped by their CDR3 sequence. So those sharing the same CDR3 sequence will be presented together as a set. And if they have other differences, they'll be shown separately on the same page. So you'll see all their differences. And we have as much detail as the authors provide. If they give us both chains, we have information on both chains. If they give us the CDR1, 2, and 3, they will show up here. And it's not that uncommon for a receptor to bind to multiple epitopes. So we'll provide for you every epitope that this CDR3 was shown to bind to. And these are cumulative, so the same receptor may be described in multiple papers by different authors as binding to different things. And then the very last results table is the references. So some people use the IDB just to see what papers describe something. So if these were all the papers that were describing coronavirus that we have data for, so this 265 references, you can export this and have a list of papers that describe coronavirus epitopes. And here, just like on the other tabs, we have the sortable column header. We have the same funnels. We have link outs to the PubMed to see those papers. And then we have the details. And this is the stable assay reference details ID that takes you to details about that particular reference. And it'll summarize all the data for each reference. So after all of that, those are all the results tabs that you can go through, and each of those tabs has their own export that exports information from that tab. We then have additionally more filters that are not available to you on the home page. And that's because we put the most commonly used filters on the home page, but we don't want to overwhelm you with lots of different confusing choices. Here, often it's you get to see your results and now you want to further narrow them. You're interested in some particularity about your results page. So here, this is additional ways you can get to it. So in the epitope search pane on the results page, there are a few extra things added in there that were not on the home page. The first one you will see is that for the non-peptidic epitopes, you can now search for particular ones. You can just type in the box. If you're interested in penicillin, you can type penicillin and it will auto-populate. Or you can click open that finder and you can see the non-peptidic tree for all of the non-peptidic epitopes we have in the IDB. And this tree originally came from the CABI ontology, the chemical ontology, but we put a great deal of effort into streamlining it and simplifying this tree to be more immunologist friendly. So originally it was made for chemists, and it had a lot of terminology that was sort of foreign to immunologists, and it had a lot of nodes that we didn't necessarily need. So we've removed intermediate nodes. We simplified the terminology to make it easier to navigate, and we hope that it's much more usable for immunologists. Then additionally, we also added the new chemical entity by role branch, so that now you can search for non-peptidic epitopes based by the role that they play, whether they play the role of a drug or a dye, for example. Those applications and biological roles can be found in that tree. And the next new thing in the epitope box on the results page is you can now also search for all data that has a 3D structure. And I'm going to show you how to do that. So once I've clicked it, it appears up on the header bar of my pending filters, and I have to hit search to get it to be applied, because the search doesn't know if I'm through adding other additional new variables. But I want to show you before I do that, there's one more way to get to 3D structures, and that is that top blue header bar that you saw on the home page. There's a browse by 3D structure option there, where you can look at antibody, T cell, and MHC 3D structures through a tree view. The B cell is organized by the organism, and the T and MHC organized by the MHC molecule. But I searched using the result page, and once I apply, I'll see rather than 3,000 epitopes, I have 49 epitopes that have 3D structure in the IDB. And when you go to the assay tab, this is where you can interact with that 3D structure. This little cube that has 3D on it opens up the 3D viewer, which looks like this. And the 3D viewer has a number of different options. You can click things on and off, and you can look at different types of residues and contacts. And if you haven't used it before, and this is something that interests you, I suggest you try it out and play with it, and then ask us questions if anything wasn't obvious or intuitive. And then the last new thing that's added to the epitope search box when you're on the results page is the ability to look for modified epitopes. So modified epitopes have some modification on any particular amino acid in their sequence. And you may be interested in all modified epitopes, and you can select all, or you may be interested in a very specific modification. And you can combine this search with the you can put in a peptide you're interested in and find out if we have any data for that same peptide being modified. Or you may just look for all epitopes that are citrinated. You can do that through here. And then the next search box down on the results page is the antigen. And this is the same as it was on the home page, so there's both organism and antigen. But we've also added here individual finders, so that you can click open these finders and navigate through them when you're on the results page. And this is what it looks like when you've opened up the antigen finder. So this is showing all the proteins in a protein tree. And I'm illustrating how insulin here has now 2 process chains available through that UniProt reference proteome that I mentioned earlier, and how UniProt shows you the process fragments broken down further. And you can select one or the other this way. And then on the results page, we have a whole new search box called receptor, which is not found on the home page at all. So if you're interested in antibody and TCR sequences, this is how you can search for them. And you can search for all data that has a receptor sequence just by using the checkbox. Or you can search for specific types, BCR or TCR, heavy light, heavy, alpha, beta, etc. You can search by chain type. If you're interested only in the beta chain, you can do that here. And you can also search by regions. Just the CDR3 region is what interests you. You can choose that there. And if you have a sequence yourself, if you've sequenced some antibody or TCR and you want to see if there's any epitopes already shown to be recognized by that sequence, you can put that sequence in. And just like with the peptide search, you can do different identities. So someone else may have found a very similar CDR3 that binds a slightly different epitope than you're expecting to get to those other sequences that are similar, you can lower your identity threshold. And if you search, and if you have sequenced an entire TCR sequence and you want to put it in, you can use the substring to see if we have any receptors that we have that fall within your larger sequence. And then on the results page, in the assay search, you have the same choices that you saw on the page, being positive, T cell, B cell, and MHC. But here we provide each finder separately, so you can open the finder individually. On the homepage, it was one finder that grouped them all. And this is the last filter on the reference page. It's a new one that was not present on the homepage. It is for the reference itself. If you have information about the reference, you want to see the data for a particular author, a particular paper, or you want to narrow your results to a time range, particular years, you can do that here. This is another place to search for the PubMed ID besides the identifier search I showed earlier. You can choose a journal article and then you can enter the PubMed ID. So that was a lot on the search, all of the different tabs and all of the different ways you can search for the data. Now we're going to actually look at some data. So the user submitted query was about COVID-19 immune responses in the IDB. And all of this data that I've shown you so far was for all of current. So now I'm going to show you how we can further narrow it. So I will go back to the organism box, and I'm going to X off all coronaviruses, and I'm going to use COVID-19. So when I start typing COVID-19, it brings up SARS-CoV-2 being a synonym, and then I select that. And now that I've made a change to my query, the search box becomes green, letting me know I've added a new parameter that I have not yet applied. And that's the only parameter I'm going to add. So I click. And then now, instead of 3,000 epitopes, I have 519. To say that we're curating the new COVID-19 papers as fast as we can, they're our highest priority. We look for them every 2 weeks, and we try and get them to you as soon as we possibly can. So these numbers are growing very quickly. And these references about COVID-19, they're all from 20, as you can see, as this is now a new important topic. So a related user query was, how can we use the IDB to have data that's publishable in a high impact factor journal? So I'm using this example of a paper that uses IDB, and Dr. Fauci is holding it up to being very important. So I think that is a very high priority, and not to mention that it's a Cell paper. And we are lucky enough and honored enough today that we have Alba here to talk about this paper and how she used the IDB to accomplish this level of important impact factor journals. So she will walk through the details of how to IDB for her project. But I want to show you, this is the paper we're talking about, and we have both Alex and Alba here, and we're lucky enough that they're very familiar with the IDB, and not only do they create data that goes into the IDB, they know how to use the IDB. And so many people use the IDB in their research, and if you look in the methods sections, you can see all the different ways people use the IDB and apply it. But originally, you're going to need a good idea. That's a good starting point. So once you have a good idea, the goal of the IDB is to help you execute that idea as quickly as possible, save you time so that you don't have to read all of the papers that go into your idea. That you can use the IDB to formulate what peptides you're going to work on, how you would formulate your vaccine, all of that. And we have a lot of use cases of the IDB. On the homepage, it was pointed out earlier that there's a use case about tuberculosis that you can click on the homepage, and there's a video walking you through that. But there's many more use cases of the IDB. We've been cited more than 2,500 times where people are using the IDB in the research. And we're working on getting these citations available to you, probably through the Learn More link, so that you can read other use cases of how these IDB. But I think Alba's talk later will really be a great example that we're very proud of. Another user query. It's quite straightforward. They're interested in looking at epitopes from RNA virus. So on the homepage, in the organism box, to get epitopes from RNA, you just type RNA and RNA virus. It's the very first hit. So once I ran that search, I get that there are 5,300 epitopes from some RNA virus. And by looking at the organism column, you can see what sorts of RNA viruses Vita 4. And as you might guess, most of the data is influenza A virus because it's so heavily studied. but we also have influenza B virus and many more different strains of virus, which is completely reflective of what people are researching. Oh, I guess I'll say one more thing about this. Once you've done these queries, you can export the results and analyze them as you see fit. And if you need to further narrow, because your question is just about the human response to RNA virus, for example, just add the human was host and rerun the query. And this is another simple but big query, MHC binding data. So we have lots of MHC binding data in the IDB. And if you want to look exclusively at binding data, just remove the T and B cell checkboxes, and now you'll get all binding data. But I want to point out we have two different types of binding data: directly binding assays, showing the binding of an MHC molecule to an epitope. It could be competitive or direct binding. It could be cellular or purified MHC. But we also have MHC ligand elution assays where epitopes were naturally processed and presented by antigen presenting cells. So if you want to exclude those ligand elution assays, you should click just the MHC binding box in the browser or type it in the Finder. And to run my query, before I continue with this example, I wanted to further narrow it to a specific MHC. If you hit the query now, it would give you all MHC data. I point out that we have nearly 3 million MHC binding assays in the IDB. So that is why the amount is going to be huge. So for my query, I decided to narrow it further to HLA-B27 binding data. And then I hit search. So here I get the filters that I had done my search for showing up at the top. And I'm going to use this example to show you an export. With and without IRI, these 68 epitopes were shown on HLA-B27. And when you do an export, you get a page that looks like this, and it shows you all the queries that the filters you used for your query. And it the little IDB icon rolls around while you're waiting, and then it gives you a link to where you can click the word here to get to your download. And the downloads are zipped up CSV files, and this is because if someone were to try and download those 3 million MHC binding data assays, it would take a while, and some people have been having their internet timeout while they wait for them. So by zipping them up, we make it faster for everyone. So then this is an export of those epitopes that bound that HLA-B27. So the epitope export looks like this. It's a CSV file, and I opened it in Excel. And in Excel, that lets you sort and filter and do all sorts of things with your data. And all of the exports have these two header rows. One is about the field group, and I'll come back to cases where you'll see that it differs in some cases. And then the second row is the actual assay field, or epitope field in this case. And the URL for the epitope is the first thing you get. And so this is that stable ID. So this ID will always take you to a page about this epitope. And if more data is added, it will also accumulate there. This next set of fields is what type of epitope is it? Linear, discontinuous, non-peptidic. A description of the epitope. In this case, these are all peptides. If the epitope modified, that information will be next. The position of the epitope within the protein. There's a section on that. And then if it's a non-peptidic epitope, we have the IRI, which will be the Kebi identifier. And we have synonyms, because non-peptidic epitopes often have synonyms. And then the next section on the epitopes in that export is the epitope export. This is something we're commonly asked about, so I want to take a moment to explain it. The epitope itself is 100% identical to its source protein, and it's specified by the author. Each author tells us what protein their epitope came from, and we respect that and use it. If they give us a specific accession, we'll use that accession. But if they don't, we pick one that's 100% identical and that the epitope is found completely in. So these are individual isoforms, the first two columns, one being the name of the protein and one being the stable IRI, which is the link out to UniProt or GenPept or PDB for exact protein. But then this next two columns about the epitope source, these are the reference proteome protein. So these are used to organize all different isoforms for the same protein. So all polyproteins will be grouped together using this exact same reference proteome protein. And that way you can sort and filter on these columns and group all data for the same protein, regardless of what the isoform was named in GenBank, because there's quite a bit of variability of how proteins are named in GenBank, and we can't rely on that to let you know that they're the same or not. But the reference proteome will always have the same name. And also the IRI is the link out to that UniProt reference proteome entry for that grouping protein parent. And in these cases, the epitope may not be 100% conserved in the reference proteome because it's one individual representative strain. And then the next two set of columns are about the source organism of that epitope. So epitopes that are natural came from an organism. And very similar to how we do the proteins, we have two sets of columns describing the epitope. And again, this is something we're commonly asked about, so I want to be sure to explain it. So the epitope as described by the author may be found in a very specific strain. So the one that's in yellow shows you that that epitope in that paper, the authors said they came from the specific strain of influenza A virus, this H3N2 strain. And we always respect what the authors say, and that the same epitope may be found in other strains, but each paper is curated according to what the author said. So this is the strain that the author said. And then the next set of fields is the reference proteome species level organism. So if you wanted to group all epitopes from the same species, you can use these grouping columns to do that. You can filter and sort. And some of our users are very interested in the exact isoform or the exact species as stated by the author. And some of our users are not interested in those columns at all, and they only care about the reference proteome level information. So that's up to you as a user which matters. Most of you, or maybe you care about both. Some people care about the peptide sequence and they're not interested at all where it comes from. So we put the data in and then we let you use it how best fits your needs. So now from that same query, MHC binding data for HLA-B27, this is the MHC assay export. So again, this is an Excel export from the assay tab. And you can see that the first column header is the grouping. So here we have information about the reference that this assay is from. And then the next section is information about the epitope. And then the second row in this Excel sheet in the export is the exact column name for the column you're looking at. So the first section is similar to the assay details view. And then we start with a section about the reference where this data comes. So we have all the columns that we saw on the assay details telling us about the paper, its title, its authors. And we have two IRIs here to point out. One is that assay. Click on that link, it will take you to the web page for that assay. And then each reference also has stable IRI, so reference IRI. This will take you to a link that's stable that gives you all the information from that paper. So the same reference may have data on many different epitopes and many different assays, and you can see that by clicking on it. With the assay IRI, specifically about the particular row we're talking about here. And the next section, once you get past the reference in the assay export, is about the epitope, and this repeats. All of the same fields we just saw in that epitope export are repeated again in your assay export. So when you do the assay export, you get information on both the assays, the references, and the epitopes. And then scrolling out in the assay exports, there may be many, many columns because we right now provide for you all the columns. And we've had requests to simplify that, and we are working on that, but it's not as easy as you would think because different people want different columns. So it's safest to give you everything and you can ignore what you don't care about for now, but we are working on making that more user-friendly. But for now, I've scrolled out to the assay section. You can see the top header says assay. So these are all the columns about the assay. And I want to show you how we've now broken what was an individual assay type into two columns. And this will let you sort on different criteria or filter. If you're interested in particular assay types or assay groups, you can do that here. If you're interested in assays that have units because you want to look at the quantitative value, you can sort and filter doing that in Excel. And then I want to point out what one of the IRIs look like. So assay types, we use OB ontology. So the IRI, when you're just choosing your export type, this is about the link outs to those ontologies. So these links take you to a stable web page that gives you more information about the assay type for this example. If it was a disease ontology IRI, it would give you a link out to more information about that disease. And then lastly, on this page, there is the quantitative measurement. Because this was an MHC binding assay export, we do have quite a bit of quantitative values for MHC binding data. For any assay type, if the authors give us quantitative values, we capture it. And you'll always see that because there'll be a unit that describes what type of value is being captured. And for people interested in MHC binding data, they very often want to see this sort of information. So you can then sort and filter your results to only get those that you think are reflective of the highest binding or the lowest binding or whatever it is you're interested in. This next query, this is about agents in the IDB. And so because the IDB has data on infectious disease, allergy, autoimmune, we have sorts of different data. So we definitely have auto antigens in the IDB, and there's more than one way to search for them. So one way that may seem obvious is to search for epitopes where the organism and the host are the same. So if the epitopes are from human and the host was human, these assays would be reflecting a human recognized. But I want to warn that researchers don't always do their research exactly this way. They may study humans and measure the response to a cow protein or a rat protein rather than a human protein or peptides, because that's what they happen to have in their lab. So then you wouldn't see that data if you did the query this way. Or very often autoimmune diseases may be studied using mice. So it may have been a mouse recognizing a human protein. So again, you wouldn't get that results if you did it this way. So I'm going to show you another way you could do it. If you're interested in a very specific autoimmune disease, you could do all humans having an autoimmune disease or narrow it further to your precise disease. And then you will get autoimmune data that way. And I want to point out too, we have all the animal models of disease. And because we use the synonyms, you can search for EAE and you'll get experimental autoimmune encephalomyelitis. So you can search for both the humans having disease, animals having autoimmune disease. And this is another query. And they're asking how they can look at allergen peptides binding to two molecules. So there's more than one way to get these results. And I'll work you through my first example here is I chose a specific allergen, Timothy grass, and then I started with the class 2 search. So when you do this search of epitopes from a particular allergen, and the restriction is class 2, I want to point out that you also get T cell data. So many people are interested in MHC binding because eventually they're really interested in T cell assays, not the MHC binding. They're just using that as a quick way to see what might be a potential T cell epitope. I want to point out that the IDB, because we have so much data, we may already have T cell data for the epitopes for which you're interested in. So first check, do we already have a T cell response tested for those same peptides? But if you truly want to just look at MHC binding data, this is how you can do it. Take off the BNT and put that you want precisely MHC binding data. So now this is that same query, the Timothy grass epitopes, but only for MHC binding data. So now the T cell tab is 0. I've removed those epitopes. So these are all the Timothy grass epitopes tested for MHC binding. And here you can see all the different types of MHC for which we have data so far. And then here also the quantitative measurements, which are very important for MHC binding. And I'll also point out that you can see these are very often the same tested for different molecules. But all of the data, and then based on the type of and how positive or negative it turned out to be, what is the best binder or the not best binder. And this another way, look at allergen binding to class 1 or class 2 molecules. So rather than specific allergen this time, I'm picking allergic diseases. So in MHC restricted responses or the context of allergic disease, then you do the query this way. And here for class 1 epitopes, we only got epitopes, and quite a few of them are non-peptidic epitopes, drug epitopes. if I repeat the query, but now I'm looking at class 2 epitopes recognized in the context of a host with the allergic disease. Way more epitopes, 4,000 epitopes, and peptidic epitopes. We reduced a few drug allergen epitopes. So based on how you formulate the query, you get quite different results. And this is another user-submitted query for how to use the IDB for vet. So it really depends what sort of vaccine you're interested in making, what patent are you interested in. Vaccine, you might want a T-cell response. Are you interested in eliciting both B-cell and T-cell responses or only B-cell responses? It totally depends what your context is, but I'll do a few. So for influenza A virus, I'm going to make a response vaccine. So I looked at what are the antibody responses to influenza A virus. So when I did that, influenza is very highly studied. So these are the 15 antigens we have for influenza. And as you might guess, hemagglutinin is very well studied. 883 epitopes from 284 different publications. So we're always adding more influenza data. So what I want to do next is look at the immunome browser. So here, this also answers another one of our user questions about how can I tell where there is both an antibody and a T-cell response on the same protein. So here, there's the immunome browser for hemagglutinin. So you can see my query at the top. And the first graph shows you the B-cell response to hemagglutinin. And the bottom graph shows you the T-cell response to hemagglutinin. And you can see the antibody, the blue line is the positive antibody response at the top, which is quite peaky. And then the bottom graph showing you the blue line for the T-cell response. There's just a few key peaks, and you can see where these two graphs overlap. So you can see the regions that have both an antibody or T-cell response, or the region that has a B-cell and not a T-cell, and vice versa, when you compare how these graphs look based on your query. And then I took it a little bit further. Some vaccines, people want just a cytotoxic response. They don't want a class, or they just want CD4 response, or they just want a CD8 response, or you want both. So again, you can use the immunome browser to see for this particular protein, in my examples, hemagglutinin, how the class 2 response and the class 1 response differ. So they definitely have their blue peaks at different locations for the different types of response. So depending on what type of vaccine you're interested in making and what type of immune response you want to get, you can compare all of the immunome browser graphs and see where they overlap and where they don't, and keep in your vaccine what gives you the response you want, and don't put in your vaccine something that's going to give you a response you don't want. This is another user requested query. And how do we look for cross-reactivity for similar epitopes? So what makes an epitope similar is up to you. You may want highly identical, or you may want similar viruses. And the example, I chose two different ways of doing this. My first example was Zika and dengue, because I knew that there were cross-reactive epitopes in the IDB between Zika and dengue. And I chose to go about it by picking epitopes that are from Zika. So I picked the organism Zika, and the host had a dengue disease. So if the host had dengue, did they recognize the Zika epitopes? So I chose those two parameters and hit search. And here you can see these epitopes are all from Zika virus. And then on the assays tab, you can see in the immunization comments, all of these hosts, these were humans who had dengue virus, and they did recognize Zika epitopes. Another way of looking at cross-reactivity is this is between self and virus. So for epitopes from humans, how often were human epitopes recognized in humans who had a viral disease? So I picked the disease was virus, and the epitopes were from human. I hit search, and here you can see these are the epitopes that are from humans. And in the assays tab, you can see the immunization was these humans had a virus disease that led to the recognition of those self epitopes. And this is another user submitted query, and their question was about Sarcocystis neurona epitopes in the IDB. And I'm sorry to tell you, I didn't find any Sarcocystis neurona epitopes in the IDB. If you know of publications that have them and we don't have them, let us know. I know that this is a veterinary pathogen, so it maybe those journals aren't in PubMed. That might be something for us to know. So I chose a different pathogen in cattle, so foot and mouth disease. To look for the antigen, I chose to type foot and then brought up foot and mouth disease. And then I chose the host was cattle because I wanted to keep it something in animals. And then what does protective mean? That is an interesting question to be asked. So different users and researchers will have a different definition of what protective is. But I want to show you using our assay types as finder, you can decide for yourself what you want to include or exclude as being protective. So opening up the antibody assay branch, using that finder tree, I can show you that we have all sorts of different in vivo assays, and some of them may indicate protection, like an in vivo disease assay. But I want to point out we also have disease exacerbation. So for autoimmunity, when you inject an animal with an epitope, you may induce a disease. So you wouldn't consider that to be protective, but you would consider decreased disease or decreased disease after adoptive transfer, which is where you put an epitope-specific antibody in an animal and it protects them from disease. So that's definitely an indication of protection. But many people also consider neutralization as an indication of protection. Some people consider ADCC or agglutination as an indication that it may be protective. So you should look through our tree, see what we have data for yourself, what you consider to be protective. And for some species that are very highly studied, you may get to be really choosy. But if you're interested in a pathogen that's not very highly studied, you may have to in your description of protection, just data has been accumulated so far. So I did this query and I got epitopes from foot and mouth disease that were shown to be protective in cattle based on the assay types that I chose.
Hey, Randi, just 3 minutes to go.
Okay, I think this is my last one.
Perfect, thank you.
So query asked for analysis of Ebola virus. So again, very broad question. So in the organism box, you just type Ebola, and then you get all of the epitopes we have in the IDB that were positive from Ebola virus. And it's up to you to do with this information what is important or interesting to you to formulate your own decision of what an analysis means. So these are the antigens. The envelope glycoprotein has the most epitopes. It's most well studied. You can look at the immunome browser and see if this gives you all that what you consider to be an analysis, which regions of the glycoprotein are positive or negative. Then you can see we have both antibody and T cell data and MHC binding data also. And then this is the export of all of the antibody assays for Ebola virus. And I wanted to just point out that once you've done that export and you have it in Excel, you can then very quickly, by clicking on the filters, do a summary of what the data is to date. So these are all the hosts that we had data for, those Ebola virus epitopes. I just filtered on the hosts in Excel. And then these are the immunization processes. Some were mice immunized with something, and some were humans who had Ebola virus. And then these were all of the assay types for the antibody export. I just did a distinct to look at which assay types we have data for. So your concept of what's a full analysis totally depends on what's interesting to you. And we do have receptors. They're all antibody sequences for Ebola virus. And then these are the references. Your full analysis may just mean what are all the references that describe Ebola virus epitopes. So you could take this and read all those papers on your own if that's what you consider to be an analysis. Oh yeah, and it was my last one. So now we're ready for question and answer.
Thank you so much, Randi, for that awesome presentation. I'm going to give you a few minutes, have some water. That was, 60 minutes. And I'd like to invite Bjoern to the Q&A. We do have quite a few questions that have come through during the last hour. So while you take a look at the Q&A box, Randi, Bjoern, if you'd like to come back online and kick off with one of the first questions.
Right. So I hope you can hear me. So one question, the first one is, what time can we expect a cancer epitope database? So the actually, as was said originally, the original contract for the IADB has not been dealing with cancer, apart from some exceptions, but there has been, as you all know, a huge resurgence of interest in specifically in epitope-specific T-cell responses for cancer in the light of checkpoint blockade, and etc. So now the National Cancer Institute has given us some funding to do a pilot curation of data on cancer-related epitopes, and those are being added to the IDB, and it's looking really exciting. But I have to stress that's a 1 year and limited amount of funding. So we have in parallel also put in a large proposal to really do cancer-specific database, to allow cancer-specific queries, development of cancer-specific tools, etc. Because we know actually that a significant fraction of the people working on the IDB are using it for cancer. So that was a long way of saying it. So we are hoping maybe in the summer of 2021, we have a yes, but so far there is data in the IDB. I encourage you to look at it, but it's definitely not complete. Randi?
Okay, I saw a question on in which disease would transplantation antigens fall? Is it okay if I share the screen again?
Of course, yeah.
So transplantation is one of the, oops, sorry, I'm clicking over infectious disease. So transplantation-related diseases are alloreactivity and rejection of transplanted organs and tissues. And that's if the host, if the epitope being studied in the assay was from a host who had rejection of their transplanted organs and tissues, or they had an alloreactivity, then that disease would be covered. But if it was the donor and they were just showing that they could respond to, say, an HY antigen or carbohydrate, that would be a disease assay per se, if that wasn't the context in which it was studied. So it's, if there was a transplant disease, it's going to be from this branch.
Okay, so I'll take the next one. It said, you indicated that you do not include HIV epitopes because Los Alamos does that. My question is, if one searches for peptide sequences and happens to represent in an HIV epitope, will IDB make that match? So the short answer to that is no. We have actually been working with the Los Alamos team and exploring some kind of integration, but there's quite a bit of discrepancies in the way the databases are structured. And so unfortunately, at this point, there is no plans to make that happen. So unfortunately, if you're mainly interested in HIV, well, that's not unfortunate. Then you're well suited and you'll find the data in the Los Alamos database. Obviously, the biggest problem is people who do kind of crossover research, which are currently just not served. So you would have to go to two separate databases.
Okay, I'm going to answer the question of, in the description for epitopes, is automatically generated or written by hand. In the epitopes details, when there's a little description of textual description of the epitopes, that is automated. It is made up by accumulating all the information from all of the papers described epitope, and so it's read a week based on if we've added data or not. In the epitope details, there's comments. Those are free text comments written by the curator for that specific paper, but on the epitopes detail is automated.
Thanks. The next one I have is on the Immunome browser, response frequency is specified lower bound and upper bound. I missed the meaning of the classification. Could you re-explain, clarify it? Thanks in advance. So essentially, what we are using is the frequencies is based on something like you've observed responses in 10 assays out of 100 performed. And that would give you a 10% frequency. And then you can use essentially a Poisson distribution of asking what is the confidence that if you had a million assays, that it would still be 10%. And that gives you the lower and upper bound, which is like a 95% confidence interval, that if you had a lot more data, that it would fall there. And as you can imagine, if you have 10 out of 100 assays, your 10% estimate is probably pretty good. If you have 1 in 10, it sucks. So obviously the confidence interval gets smaller and smaller the more data that you would have. And there's a detailed write-up, I believe, linked to, or at least there should be, on the Immunome browser page that goes to the math specifically.
There is. There's a help desk topic. Okay. The one I'm going to answer is, there was a suggestion that if the method of the paper was included in the summary. In the details in the immunization section, if there was like an administration, we do capture a great deal of detail on the route and the dose, adjuvants, things like that in the immunization section. On the assay itself, we capture captured the assay type, and then any of the details would just be in free text comments. And hopefully that's enough detail for most people, but we do link everything back to the paper it came from if you really do want to see differences in the methods. Minute differences in the methods is really beyond the scope of what we can capture, but if there was anything interesting or strange, the curator would comment on it.
And as a minor addition to that, we would actually love to just have the full text present of the paper and highlight this is where it's described. But then we get into copyright issues. That's also why we can't copy over whole paragraphs of text. So yeah. Next, I have receptors tab. 2 of the headers were chain 1 CDR3 and chain 2 CDR3. Which chains do these each correspond to in CCRs and BCRs? I'm assuming chain 1 is the alpha chain and 2 is beta chain in TCR. Is that correct? How about for antibodies? So as you saw, I think the question was asked before Randy showed the detailed query interface. You can search for specific types. So for T cells, yes, chain 1 is normally the alpha and chain 2 is the beta, except for if you look for gamma delta TCRs. And so for antibodies, similarly, normally chain 1 would be light and chain 2 is heavy, maybe, or the other way around. Check the query interface by what the breakdown is, but then you have like weird animals that have like heavy chains. You have nanobodies which only have a single heavy chain. So that's why we don't have like a header there, because it's going to be, it could be different for all kinds of subtypes of data. So essentially, either just limit the search specifically for alpha beta TCRs, then you know what chain 1, chain 2 means, or you have to examine a little bit more detail.
For the next one, I'm going to take just screen share. The question was where to find the pending filters. So I need to put in some choices so we get some pending filters. So I've made some selections, and when I hit search, my pending filters show up here in the header. These are the current filters. And the pending filters is now on the results page. All of these are filters you could choose. And if you chose one, it would show up on the header row. So all of your filters for your query are up at the top. And if you X them out or change things, the search box turns green to let you know that what you're seeing doesn't reflect what the filters are.
Okay.
I think that covers it, right?
I think so. I think the next one I have here is thank you for the demonstrations. Is there a way we look more broadly at the genus to find promiscuous epitopes between different species for microbiology studies? So yes, there is. So you can, essentially we have the whole NCBI taxonomy tree available for queries. So you can search this for all bacteria. You can search for actinobacteria or mycobacteria, or, and this is the tree kind of going down: bacteria, actinobacteria, mycobacteria, and then mycobacteria tuberculosis complex, mycobacterium tuberculosis specifically, and then Mycobacterium leprae, and all the other things in there. So you can query at any kind of level of hierarchy, and it'll include all the organisms that are subsumed by that query.
Okay, I'm taking the question of, are the selection criteria added to the export? This is a really good question because we're doing that now. Right at the moment, today, no, but on the test site, yes. We are already working on this because it's something people have asked for. And as an aside, we're always adding new things that people ask for. So if you'd like something, now is a good time to give us feedback. You may not see it in a year, but it will still stay on our list of things to do. We work through the ones that are requested the most frequently first, the ones that would make the biggest impact. And this is one that's been requested more than once, so you'll get it soon, within the next year.
So the next question is also a very good one. When I search for, I mean, all the questions are good, sorry, but we like the ones the best that we've been thinking about and are working on. So when I search for a specific TCR, after I click on receptor tab, I get information about CDR sequence, gene usage, epitope, beside other informations. Would it be possible to also include information about the MHC of these epitopes in this session? Absolutely. That's been coming up several times now, and we are working on it. This is not something that's going to be coming up in the next release or so, but I'm certain we can have it done in the next 6 months or so. I'm hoping. It's actually I think that's a big oversight on our part. I mean, the database, as you see, is complex, so that's one of the things that we missed.
Okay, I'm gonna share screen again. There's two questions in a row. Bjoern, I'm overstepping. Two questions in a row that are sort of visual. One was about the assay types for MHC binding data. So when you open the finders, you see all of the assay types for which we have data, and you can click through all of them. And so if there's a particular experiment type that matters most to you, you can select more than one. These are all multi-select, and so once you pick them, they show up. And once you're done making your selections, I can't see because of my screen, you hit the apply button. Whoa, I did not want to open it again.
And I'll just say we're at time, so we'll finish with these last two questions. Thank you.
Okay, well, the next one was just visual. They had asked where you see the immunome browser. So it's on the antigens tab. And when you first go there, it'll be popped up to highlight that it's this little graphy icon. So when you close it, the immunome browser, it works per which protein you want to apply your epitopes along. So every protein on the antigens tab will have that little graph next to it, and that opens the immunome browser. Okay.
So then I think we have to stop here, and I'll just, maybe Randy, we can type some answers in for the remainder of the questions during the session or so. And otherwise, if there's things that we can't answer, obviously, feel free to stick along to the last optional Q&A. If there's more complicated things you want to discuss, that would be a good opportunity. Thanks, guys.
Absolutely. Thank you so much, Randy, for that presentation, Bjoern for joining. And as Bjoern said, we will not leave any questions unanswered, either via the chat or right at the end of the day.